used to view the world as a place where I gave the word. It had the feeling of eminence. Then along came evidence. I used to give advice, see disarray in my patient's eyes. I listened to the company sing. Now the old drug is dead, we got anything. 1 minute I held the key, next the walls were closed on me, and I discovered that my practice stands upon it.
So I just want to introduce Dr. Benjamin Eaton, and he's from UTHSCSA. He's an assistant professor in the Department of Physiology and for the Barshop Institute for Longevity and Aging Studies. He earned his BA in his native Colorado and then saw the light and moved to the proper state of California and got his PhD from my alma mater, Berkeley, and his fellowship training, or his postdoctoral training from UCSF. And he and his lab are now interested in defining the molecular mechanisms that stabilize and maintain synapse function throughout the lifespan of an organism. They predict that these mechanisms contribute to age-dependent declines in nervous system function that are observable normally and during disease. And their work has been published in Aging Cell and Journal of Neuroscience. Thank you very much, Dr. Eaton, for coming.
Yeah, great. Well, thanks for having me over here. Try to make this as informative as I can. So, do I need to stay out of the way here, or?
Anywhere you want to go works.
Okay. I want to make sure everybody can see.
That's probably the best spot over there.
Over here somewhere. Okay. So, thank you. In the lab, we are interested in, as was mentioned, synapses. Okay. Today, I'm not going to talk too much about synapses. A little bit. I'm going to give you a flavor of some of the work that we've been doing recently. I think that you might find interesting. I'd love to get some feedback from all of you on this. Okay. It's sort of taking me in some place that maybe I don't have very much experience with. And in some levels, I think where we're headed is frailty. Okay? And I don't know if we've gotten there yet because, you know, frailty is one of these things that maybe you guys can help me with, because I think you know it when you see it, but I'm not sure if I know exactly what it means. Okay? But certainly I think one of the things about aging is there becomes a point in time when you can look at somebody and say they're frail for whatever reason that might be. Okay? And I think some of the stuff we're talking about today may impinge upon that, or we may be finding some ways of trying to improve frailty. Okay. So there's a number of things that, in terms of the nervous system, appear to decline in terms of function with age. Okay, so there are obviously declines in cognition, and there's also declines in motor function. And we believe that most of the, well, it's clear that for both of these, there are central control mechanisms, brain, nervous system functions that contribute. Okay? And the kinds of things that you also see in terms of changing in motor function, in terms of things like strength, stamina, and control, we also believe can have a presynaptic component, a neuronal component. Okay? I guess these are the kinds of things that I wonder about, whether things like strength and stamina actually are part of frailty or not. Okay? And we think that, and there's good evidence to suggest that at least some of these declines, there's a contribution of synaptic dysfunction. Okay? And that synaptic dysfunction would be age dependent. So there's some deterioration in synapse function that we think at least is at least a partial contributor to some of these declines. And in the lab, we want to try to understand the neuronal contribution to age-dependent declines in motor function and synaptic function. And what you're not seeing here is that we would like to determine the cellular mechanisms that are responsible for these changes. And so this is the model system that we're using for these studies. So it's Drosophila. And what you're seeing here is this fly is doing what we study a simple motor reflex, and it is a reflex. That is, we are generating this by stimulating the sensory receptors on the. I'll have a pointer. What you're not seeing is over here is the foot of the fly. We're stimulating the foot of the fly with sugar water, and this fly has been deprived of water and food, and under those conditions, you get a maximal response, and it is a reflex. That is, you can vary the stimulus here and get exactly the same robust response under those conditions. Under other conditions where you haven't starved the animals or they're not, and they're satiated, you can actually get differences in terms of this behavior based on the strength of the stimulus. Under these conditions, there's no change in the behavior when you change the strength of the stimulus. This is a young animal, and what we find is that this is a behavior that declines with age. Okay, so, and what I mean by declines by age, I think you can tell that this extension event, oh, well, it's turned off. I think you could appreciate that it changed. Okay, so one of the things about this behavior that there was a couple of reasons why we've chosen this behavior. One was, it was a reflex, okay? And not only that, we felt like it was a very specific motor function that we could be very quantitative about. Because what we could do is we could make a movie of the event, and then we could go back and draw paths that a individual sensory bristle traveled as it went through space. Okay? So there are sensory bristles here on the tip of the proboscis, and we track those over the course of the movie and can generate a path by which the proboscis extension occurred, and that allows for quantitative analysis of things like velocities, displacement, which is the linear length under which this moves, and aspects of how the shapes of these paths might be changing. Okay? And I think you can appreciate that at the level of sort of path analysis, you can sort of appreciate that there's a sort of a greater spread in terms of what types of extension events occurred, potentially indicating some kind of a deficit in motor coordination. But for this talk, we're really going to focus on these, and particularly this velocity measurement is sort of our measure of motor function. Okay. And so if we do path analysis of flies across lifespan, what you can see is that there's a consistent and progressive decline in the velocity at which these animals stick out their proboscis. Now the size of the proboscis doesn't change. It's very consistent from animal to animal. So this is not due to a change over time with the proboscis size, or a change or differences between individuals. And even this oops, keep pushing the wrong button. We even find that sort of interesting that there's an abrupt change in how far the animals can actually extend their proboscis between 35 and 42 days. So in terms of the lifespan of these individuals, a 63-day-old animal is about at its 85th percentile of life. At this point is sort of our end of where we like to do analysis, because these animals have a very consistent survival curve, and so once you get past this, you kind of have this sort of change in mortality that you see at the end of lifespan. I try to stay away from this. We generally don't go past about the 85th or 90th percentile in terms of these behaviors. Now, as I mentioned, we're interested in understanding the neurological contribution to these changes that we observe, if any. Okay, so we don't know a priori whether or not there's any sort of neurological sort of contribution to these changes. One thing to remember is that neuromuscular junctions at flies as well have safety factors. Right? Safety factor is the term that says that this synapse here releases more neurotransmitter than is required to depolarize this muscle. All neuromuscular junctions do that, and they vary from different muscle groups. Okay? It's a safety factor built in because this is a particular synapse that when it fires, it has to fire. You have to depolarize the postsynaptic cell. That's a difference between neuromuscular junctions and synapses in the CNS. The CNS synapses fail. Neuromuscular junctions do not. And I think that's probably evolutionarily built in so that when you need to run, you can run. And we got particularly interested in a neuromuscular junction that is situated on one of the muscles that's involved in that extension event, okay? And that's one of the reasons we were interested in that motor behaviors, because we were interested in studying this neuron, and this neuron had some real benefits for us in terms of doing these studies. Okay? One was, there's about 12 muscle groups, maybe 13 muscle groups that are involved in this behavior. Okay, so this is just one of them. But it had been previously established that this muscle group was required for a normal event. Okay, so if you don't get proper depolarization of that muscle, it does read out as a defect in the behaviors that I was just showing you. Okay? The other thing, which was even more fortuitous, was that a group in Berkeley had actually found an enhancer unit that allowed us to express genes specifically within this motor neuron and nowhere else. And so now we had a system where we could specifically manipulate, and I should say there's another neuron over here innervating this muscle. I'm just not showing it for ease, and it is also driven, or it's also under the control of this enhancer unit. And that allows us to drive whatever sort of genes or mutations we want in this neuron, look at the behavior, and know that the only manipulation that we've done in the animal is to manipulate these neurons. So we have a real nice cause and effect relationship, and that's one of the real reasons that we decided to pioneer this. So I should say no one has ever did recordings from this or looked at this synapse before. Okay? And so we had to do quite a bit of work to get through to the data I'm going to be showing you. Okay? So we pioneered a good dissection. So just to remind you, obviously you know this isn't life size. But remember, I want you to just think about the size of that fly, that fruit fly's head. And I think you can understand that these are pretty difficult experiments. And the person that is probably the best in the world is sitting there. That's Rebecca. She's my graduate student. She is amazing at doing the recording. So there were two things we had to do. First of all, we get to see if we could look at these synapses. One of the things that we want to do is be able to image in live time, look at things like calcium imaging. So we had to be able to dissect and get a dissection where we could see the presynaptic nerve terminal, and that's what this is. So this is the presynaptic nerve terminal of this motor neuron, and it's making this ramified innervation onto this muscle group. So this is the sidereal 9 muscle group, and it consists of about 15 muscle fibers. This synapse has a number of the markers that we have reagents for, so we can look at the presynaptic side by expressing a GFP in the motor neuron using that driver that we have. We can stain for postsynaptic proteins. So we can analyze aspects of innervation. And we have lots of different tools that allow us to look at very high-level resolution at individual, so these are actually individual active zones. These are actually release sites for synaptic vesicles. So we have a real high resolution as well as a number of tools that allow us to really dissect what's going on at this synapse. And it's very stereotyped. Right, so you can go from animal to animal to animal to animal, you get the same number of innervations, the same number of muscle fibers, and even the area is very conserved. Okay, so it's a nice system that we can take and start teasing away things genetically, which is sort of how we like to do it. Importantly for today, and this was a major breakthrough, was being able to record electrical activity at the synapse. Okay? So this is our setup. Oops, sorry, pushing the wrong button. We come in here and stimulate the nerve here in what we call an en passant stimulation protocol. And we record the resulting depolarization of the muscle with a recording electrode that's impaled in the muscle. And what you see is you can evoke a response by generating an action potential, and we see that as a large EJP, end-plane junctional potential, or end-plane excitatory potential. We can also see the spontaneous release of single vesicles, and that allows us to analyze the number of vesicles released per action potential, which is a very powerful way of understanding the strength of the release event and the strength of this synapse. Okay? And the way we do that is simply, we can take the size of these miniature vents and compare that to the size of the evoked event. And actually, if we take those two values, so we take the size of the evoked event and divide it by the size of this miniature event, we can get the number of synaptic vesicles that are released. And that's our assay for the strength of the release event. And this is exactly the amount, required to depolarize this muscle, as it turns out. And one of the other things that I should say before we move on is that this value, under the conditions that we do, which is a relatively low calcium, is really a measure of the probability of release at this thing. And so, we had already looked at the behavior, and so we wanted to know what was going on at these very same synapses at the same time that we saw the motor function declining. And what we saw was something that we were a little surprised to see. First of all, we saw a very stable response over age, and then an abrupt change between 35 and 42 days that was relatively maintained out to 60 days. You can see this is down a little bit by 60 days, but it's significantly different than that as well. So there was this abrupt change. So we didn't know what to expect. I guess maybe a priori we might have expected that the release would have dropped down considerably, but it didn't. This is quantifying that. Okay. And one of the things I wanted to point out is that this event does not coincide with changes in the number of innervations or even changes in the total synaptic area. Okay, so what we're seeing here is not that the synapse is growing, but that at this event, decides to release nearly twice as many vesicles over the course of 7 days. Which is quite dramatic. And remember, this is occurring during this declining muscle function, right? And so we had this relationship where we saw that our release made this abrupt increase and the behavior made an abrupt decrease, okay? And so that might suggest that there's a relationship, right? And so we decided to try to probe whether or not this change in release is a compensatory or maybe a pathological. And so and unfortunately, I won't be able to answer that question for you today, but we've tried some hints, and it's given us some sort of interesting hints as to what might be going on. So one of the things that we thought about was, if this is a compensatory response, it might suggest the existence of a signal that would originate in the muscle that would inform the nerve terminal about the contraction event. Okay? And this has not ever been shown to exist, but we knew that at this same synapse, there had already been a retrograde pathway that had been identified that was involved in what's called synaptic homeostasis. And that got involved when the excitability of this muscle was altered. And one way that would manifest itself experimentally is something in this sort of a paradigm where you take the muscle, let's move to the next one. Darn. Okay. So one way you could induce this homeostatic event is by changing the excitability of the muscle, and you can do that using a toxin called philanthrotoxin. Philanthrotoxin is a toxin that binds to the receptors on the muscle. So it's a pharmacological lowering of the sensitivity of the muscle to release neurotransmitter. And so if you throw philanthrotoxin onto this preparation, this is your evoked response, these are your mini responses. I think you can appreciate that these mini responses prior to the addition of the toxin are normal, and after you add the toxin, these miniature events go down. The miniature events are really telling us what's going on the muscle and how the muscle is responding to neurotransmitter. And if you wait long enough, what you find is that while initially this evoked response is down because you've actually lowered this event, the system kicks in and releases more neurotransmitter, bringing the size of this EPSP back up to a normal size. And that's the homeostatic response. It's called a synaptic homeostatic response. And it absolutely requires signaling from the muscle to the presynaptic nerve terminal. Now, in experiments that were looking at this pathway, there's mutations that have been identified presynaptically that block this. And so we decided to try to use one of those just to investigate whether the event that we saw was related to this event based on the fact that we assumed that there would be a pathway. And this was a pathway that was already existing, so we wondered if maybe this pathway was the pathway being utilized to change the presynaptic release. And so the Davis group at UCSF, where I used to work, had found a mutation in a gene called affexin, which is down here, affexin, and they showed that that was required presynaptically to get a homeostatic response. And this data is just that data showing that indeed when you get blue R is a mutation, so as opposed to using a toxin, in these experiments they actually used a mutation in the receptors, did the same thing as the toxin, lowered the sensitivity of the muscle, and under normal conditions, this is a genetic control, you get this large increase in guanyl content, that's the homeostatic response, and the effects of mutant blocked. And so we could simply ask, well, if we look at this event that occurs late in life, does it still occur in an effects of mutant? And it does. Okay, so the effects of mutants did not block this homeostatic response, and so we were looking at something that appeared to be different than that phenomenon. So, sorry this is a little out, but homeostasis is, there's lots of things that are under homeostatic control. These are control mechanisms that have a certain structure to them. Okay? They are in place, that the systems that are under homeostatic control have an output. It's a defined output, and that's called a set point. And the system is built to maintain that set point, so you have constant output. Okay? So there's a number of situations within the, I don't have to tell you guys about homeostatic set points, things like body temperature, blood pressure are things that have been shown to be sort of under a homeostatic control. Well, there's also homeostatic controls within the nervous system, okay? And they use the same kinds of ideas. That is, it's a combination of sensors, feedback, and effectors to maintain output. And there's a set point, and the system compares the set point to what's the actual output, and if there's an error, it responds by adjusting the output using an effector. Okay? And that's exactly what the nervous system does, and I'm going to show you examples of that. And one thing I wanted to point out is that there are very few examples of non-pathological changes to set points. So set points have been shown to change, but it's very hard to find a set point change that's a beneficial or non-pathological, except for maybe a fever. Because there is evidence that fever protects against infection. So under those conditions, maybe fever is an example where you've changed your body temperature set point, and it's a beneficial and not, I guess it's non-pathological, but most of the changes in set points appear to be in response to some pathological condition. And so there's a couple different ways that the nervous system, you can see homeostasis manifested in the nervous system. So firing rates of neurons are under homeostatic control. And so this is the firing rate of a neuron. Its neuron types are oftentimes defined by their firing rates. It's a defining aspect of their identity. And it emerges, like most of these, during development. And if you take certain neurons and artificially change the firing rate, and you can do that genetically, you can do it pharmacology, what you find is the neuron responds by changing its excitability, and actually will change its constellation of potassium channels and sodium channels to reestablish a more normal firing rate. In addition, excitability and synaptic scaling is another example where if you change the strength, say, of so here's a presynaptic nerve terminal. Let's just take you through this. This is postsynaptic spine. Here's our targeted activity. If, for example, you potentiate the synapse, causing high activity, the system will readjust by lowering potentially the input from another synapse to try to bring the targeted activity back down. Okay? And what people have found doing experiments somewhat like this is that if you take a cell and just leave it by itself, and you put on something that blocks activity, that cell will respond by putting more excitable sort of receptors on the surface. Okay? So they have a way of trying to maintain the balance of negative and positive inputs. But the synaptic homeostasis that we study is this homeostasis that I mentioned to you before, where you're able to block the sensitivity of the muscle to neurotransmitter, and the system increases release, which is indicated by these two, to get back to the normal level. Now, this set point, in this system, it's been proposed that the actual size of this EPSP is a set point. That is, the system is trying to maintain appropriate depolarization, which means you need to move the membrane of the muscle from here to here, And that's the set point. That what needs to happen in the system is you have to depolarize the muscle to the point where you activate the voltage-gated calcium channels and voltage-gated sodium channels, and that's what's being maintained, and the output is these neurotransmitters, and the set point again is this EPS. Okay, sorry about all that. But what we thought is, we wondered whether what was happening in the system, since it wasn't a homeostatic response, one thing it made us wonder was, well, maybe homeostasis has changed, because we knew that the synapse was functioning at a higher level. And so the way we do these experiments, and this is just showing you a timeline of these types of experiments. So what you're looking at here are the EPSPs, okay? And we're firing these along. If we add the toxin at this point, what you see is a gradually decline as the toxin binds up and lowers the sensitivity to the neurotransmitter. And then, over a very standard time scale, it comes roaring back to where you get an EPSP that's fairly close to what you began with. The EPS, the minis are still small, the spontaneous events are still small, and so what you've gotten here is a large increase in the release rate. And again, that's blocked by effects, and you can see that's down. Okay, so the question was, if we use that system to induce a homeostatic response, what happens if we compare the homeostatic response to this synapse to, say, this synapse? One possibility would be that if we challenge this synapse with philanthrotoxin, it would return to this level if the homeostatic set point was fixed, right? The idea is that the homeostatic set point is fixed here, and so if the homostat has not changed, when we challenge, we would expect this to come back to this level, not to this level. Is that clear? That's simply the simple experiment, okay? And what we found was that when we challenge this synapse, the homeostat works to bring it back up to this level, and not this level, suggesting that the homeostatic set point has changed with age, very abruptly. And this is just quantifying these changes. So this is both the change in the EPSPs are significantly larger. The change in the number of vesicles relieved to attain the homeostatic set point is also increased. So these older synapses go to a higher point, and they release more vesicles.
Greg, does the NMJ, does it change anatomically with aging?
No.
I mean, does it look like it's...
Very little. We see very little at our synapse. In rodent NMJs, you do see. It's not as clear in, I think, humans. You know, humans' NMJs are actually pretty small compared to, and so I don't know whether it's been completely, and maybe someone here would know whether in histology, whether you see empty gutters. These are sort of things that people have seen in rodent NMJs with age. We really see very little changes in structure. Okay? We haven't done a lot at the very, very, very end. We usually don't go out past about the 90th percentile. We haven't done much at the very end state of these animals. So maybe at the very end, things really go bad. That would be maybe part of death or something. But we see during this time scale, there's no change. No change.
Yeah, Holly Van Remen had a postdoc at Eaton. We started a study looking at humans' effect of aging and effect of exercise. And unfortunately, I think he went to New York or something.
Right.
We haven't followed it.
So I'll get to some data about what happens with age in the rodents. Okay, so there are some functional changes that are similar to this. Okay, so actually there is some evidence out there that might suggest that this phenomenon might be occurring at older synapses. Okay, and I'll show you that in a minute.
Does the neuromuscular junction function essentially the same across all species as far as like the sodium potassium channels and the types of neurotransmitters? I'm just curious.
Yeah,
I have
sure.
No idea.
That's a good question, and I have to say is that this synapse is very different than a mammalian synapse. Okay. One of the big differences is the structures. Okay, so we don't have gutters. Matter of fact, this synapse, if you look at it ultrastructurally, looks like a central synapse, and it releases glutamate. So it's actually, in terms of the molecular structure, it's probably more like a glutamatergic synapse than it is a mammalian neuromuscular junction. Other than it has this roll, right? So the rolls are the same. And so I think the consistencies between the preps have to do with that roll, which is to maintain the appropriate muscle contraction, which is very similar. So the muscles in this are very similar to the skeletal muscles. One of the things about this is that, you know, as I mentioned before, you know, if you think about a change in body temperature during fear, it was beneficial. Now, one question we wondered was, well, okay, this is great, we see this, but we also know at the same time we're seeing a decline in muscle function. It doesn't stop declining, right? And so, while we have suspected, and we still do, to some extent, that this is a compensatory response, it doesn't mean that it's completely beneficial, maybe. Right? One of the things that we were wondering about is that in a lot of situations, this type of potentiation might be expected to leave a synapse in a condition where it might be susceptible to running out of or exhaustion. And so actually, if we take these animals, we take a young synapse and an old synapse, and we stimulate them at 40 hertz, which is a physiologically relevant firing frequency, the young synapses have no problems maintaining that frequency. Okay? That means their cycling of synaptic vesicles and the release is all working perfect, and they could maintain that amount of depolarization for a long time. The older synapses, on the other hand, rapidly started to run out of vesicles, and that's what's indicated here. And if you look at the sizes that we're talking about here, you know, we've decreased the depolarization by over half. We actually estimate in other experiments that the safety factor at this neuromuscular junction is about 2. So, what that means is that this decline could actually contribute to maybe muscle weakness, because it could be that after a long burst of activity, and this is 15 seconds, this is a fairly long, if this was a feeding event, it would be a fairly long feeding event, but not out of the realm of what they do. And you can see that we get a pretty rapid decline, even within 7 seconds, which would be the kind of thing that these animals might be experiencing. Okay? So it could be that this would be responsible for maybe some of the changes. We don't have any way of knowing that. So, getting back to what you had asked me, so there has been some data suggesting this, okay? I'm sorry this is not just a little off.
Yeah, we should be able to.
Yeah.
So has anyone else characterized other motor responses other than this reflex, like maybe some other behavioral way, like
So,
fly?
in terms of motor behaviors for flies, people use a lot of different ways of looking at it. So one way is to see how fast a fly can climb up a wall.
Tall person.
I tried.
Another way people look at it now, a lot of people look at just general activity. So you take and you put a laser beam in a vial, and you ask how many times the flies move across and break it, which is just sort of a general sort of activity measure. One of the things was when
There you go.
it looks okay here, but it's okay. I will feed. This is Banker and Robbins, and I think you'll probably be able to get to this online or something. Okay.
Yes.
So we initially tried to look at some other behaviors. One of the things that we wanted is we wanted a behavior that we felt would at least reflect in some ways neurotransmission at the synapse that we were recording from. And when we looked at other behaviors, it was hard for me to convince myself that what I was looking at was that I was ever going to be able to figure out the contribution of sort of central processing of motor control versus the muscle. And so one of the reasons that we decided to really focus on that, because we could have thought about maybe looking at an arm or a leg. The other thing is those synapses are pretty complex, and they're not, that synapse here ended up being very beautiful and very stereotyped. So it's actually got a lot of benefits that the flight muscles are very hard to do these experiments with. So there wasn't really a great system. Now what that means is that I can't tell you that this happens at every synapse, because I haven't looked at other synapses. All I can say is, one thing I can say, is that in old mammals, seems to be a similar phenomenon. Okay, and so these are some old papers from, so this is Baker and Robbins. Nick Robbins was a very famous old neuromuscular junction guy. And I'll show you some of this data in a minute. Okay. But what he found was very similar to what we have. Okay, that is, in the older animals, they released more neurotransmitter, and they had a depleted synaptic vesicles at their neuromuscular junctions. And that was also similarly seen at actually central synapses in the hippocampus. Similar phenomenon. an increase in release, and an ability to deplete those synapses of synaptic vesicles. So this may be more common. And one of the ideas, particularly from Peter Land's field, is that this is all potentially due to the dysregulation of calcium in the nerve terminal, which has been a long-standing hallmark of age-dependent decline in the nervous system, is a loss of appropriate calcium homeostasis, and that these would be the kinds of things that would occur. That hasn't quite been proven, but there's a lot of strong evidence suggesting that, and it's really pointed at the calcium channels as a potential source of the calcium with the change with age. And so this is the stuff from Nick Robbins, and so these are different muscles, EDL, soleus, and diaphragm. This is the old animal, this is the young animal. Sorry you're not seeing this. This is the size of the first EJP. And so I think what you can see is, again, the older animals are releasing more, at least at these ones here. You can see this, not every synapse in the animal, okay? The diaphragm doesn't show this event. So why that is, I don't think is clear. Okay? But they also saw sort of a similar thing. That is, the older synapses, which are now here in the white, appear to have a little more sensitivity to rundown than, say, maybe the younger ones. So there is some similarities to what we're seeing in terms of what's going on at our neuromuscular junction in the fly, as well as what's going on in these old mice. So suggesting that there may be something similar. And so what we think is that this age-dependent facilitation represents a change in the homeostatic set point. And that homeostatic change might be due to something like changes in calcium channel activity. And we have some evidence that suggests that's the case. And this change would be predicted to change both the function and the plasticity of the synapse, similar to what I showed you. Okay, so this change in homeostatic set point may not be beneficial. One thing is that if we could block the event, we could then potentially look at the effect on the behavior. So without that manipulation, it's hard to know whether or not whether the costs of running down your synapse are worth the benefits of increasing the release. And it may be. I mean, I would predict that it's compensatory, that if we were to block this event, that the motor function would get even worse than it does. But we don't know that yet. Unfortunately, we haven't been able to do that experiment. But we tried a different experiment. Okay, so we've talked a little bit about this. We've talked about this ADF and the loss of motor function, and we wanted to know, in the end, we did want to know about the relationship between muscle function and release. What that meant is that we needed to know a little bit about muscle function and how muscle function might be changing at the same time. And it gets back to this question, okay? And what we knew was, so what we decided to try to do was to find a way to improve muscle function and see what happened to release in the old animals. We wanted to go both ways, but unfortunately it was easier for us to try to go to improve muscle function in age, as it turns out, because there was a drug available that we suspected would work in the fly, and it's called S107. It's now going to be coming out in a new form called Ripal. And it was developed in Andy Mark's lab, and he showed that it improved muscle function in mice in an age-dependent way. So an improved age-dependent function, muscle function in an old mouse. And it might work in humans. Okay, so as I was talking with Nick, I was trying to find a new source of this because it was pulled off the market by a big farm group. And in looking around, I found that I can't buy it, but the cyclists have it. Okay, so this drug has been taken within the last couple of years by cyclists, right? They're thinking that it's going to improve stamina, and so it's possible that this is safe to take. It has not been approved, but not that cyclists really care about that.
Yeah, exactly.
Right? And so the drug works like this. All right, so this is the normal situation in the muscle. Here's the sarcoplasmic reticulum where the calcium is, and it's released via this ryanodine receptor under a depolarization event, and that stimulates the contraction. And the ryanodine receptor has a small protein called calstabin-1 that binds to it. Okay? And with age, the calstabin-1 becomes oxidized. And when it's oxidized, it no longer binds to the ryanodine receptor. And under that condition, the ryanodine receptor becomes leaky. That causes the amount of calcium in the SR to decrease and effectively lowers your contractile strength when you get a depolarization event. And so this drug rapidly reestablishes this binding to somewhere on the calstatum. It's actually been defined where it binds. I'm just not a structural chemist. And so what we want to do is say, well, can we reverse any of this with S107? And we could. And so what you're looking at here is a 7-day treatment. So these are 7-day-old animals. They're on the drug for 14 days. And we assay animals that were either on the vehicle or on the drug. And so you can see on a young animal, the drug has no effect. But as the animals get older and older, we have a progressively stronger effect. And that's due, we believe, to this age-dependent oxidation of the target. And it improves all of both our velocity measurements with very little effect on this displacement deficit. So we're still not clear what the underlying cause of this is, but this supports that the drug is working to potentially improve muscle function in these animals as well. I should say we know the target of this drug, and I didn't put the data in here, but this effect is abolished in the mutant. So it looks like it is working very similarly to how it works in the mammalian system. And so, again, we were interested in what might happen to this ADF in the presence of the drug. And interestingly, if we put the animals on the drug at 35 days and then assay their function at 42 days, remember this is where we had this was the time scale under which our synapses doubled in their release event. It actually turned out that when we looked at the size of the release event or the number of vesicles, it was actually significantly reduced compared to the controls, which would be supportive of a compensatory result. But we don't think that this is a very strong result, because it doesn't actually get back to the normal levels. And our experiments recently suggest that it could be that there might be a presynaptic effect of the drug that we're teasing away right now. So I don't know, I still can't tell you whether it's compensatory or not. I will say one thing that we thought it would allow us to do is say, well, one of the things that we wonder about is, does muscle function and motor function have anything to do with survival in a fly? It's not clear to me, and maybe you guys can chime in on what you think, whether declines in motor function contribute to survival in humans. And I don't know whether that's true or not. But it's actually not really even clear in flies. So flies, it's not clear why flies die. And I've suspected for a long time that they stop moving and then they can't feed and they end up starving and desiccating and dying of starvation and thirst.
Well, I mean, walking speed is a very gross measure, and
Right.
but walking speed is a very strong predictor of mortality in humans.
Yeah, that's so the Baltimore studies, I've seen those.
Yes, and there was a paper by Studensky that came out in 2010 or 11 maybe, and they looked across multiple cohorts, and in all cohorts it's a very strong predictor, and they even have, I don't remember, but they even have it down to a specific cut point of how many, I think it might be 0.6 meters per second or something, but there's a specific cut point they could even identify that is discriminate for mortality. So
Would anybody, I mean, is it just, so what does that mean, do you think? I have no idea what it is.
yeah, I mean, like I said, I mean, walking speed has more than just strength. It's got, probably some balance measures and coordination there too, but
Cardiopulmonary function.
well, that too, yeah. So
Okay,
I mean,
so
it's
it's
not,
just-
but it's definitely,
Like integrated
Denotes-
measure of health.
yeah, there you
Right.
go. That's exactly what I was trying to say.
Okay. Because I do see that it's one of the, it seems like in a lot of the human studies, it's one of the main assays that's done is to have people stand up, sit down, walk across the room, and that kind of stuff. So it must be a very important, it's interesting to me that
It's
it's
that,
so.
and grip strength is also often used, but I think, well, partially because Stephanie Studensky is very prominent and she's focused a lot on walking speed, but I think walking speed has probably been shown to be more associated with
Yeah.
mortality than grip strength.
And obviously the push to validate it is that's very cheap and can be done
It's-
in any clinic
Yeah.
Right.
versus something more sophisticated.
Well, so the question would be, I mean, I'm assuming though that in terms of geriatrics, improving muscle strength in the elderly would be an important benefit for health span, or certainly I would imagine. I don't know if it would, I don't know whether it would extend lifespan, but certainly it would improve lifestyle, I'm assuming. I don't know.
I mean, I don't know, that's a good question. I would guess if you could extrapolate, if you improve muscle function, you improve gait speed, that your mortality would improve, but I don't think anyone's ever done
Right.
that study, but yeah.
But it's ongoing, right? The life study,
Oh, yeah, there is that.
it's a very,
life
it's a
study.
large, very expensive
Exercise.
exercise intervention
Right.
study that is looking at that.
Well, certainly exercise has been well established to improve neuromuscular junction sort of morphology. You know, it hasn't really been looked at how exercise might improve release of synaptic vesicles. It's not clear, but it certainly improves what the synapses look like, and it makes synapses look like they were calorically restricted. And so that data is out there. But whether or not it affects release is not clear. Well. So, I don't know about humans, but we suspected that this would extend lifespan. And so, what you're not seeing here is an arrow right here, okay? This is where we add the drug, okay? So what you're looking at is two cohorts that are on the same food. At this point, we either add this S107 drug or we add the vehicle. And what you can see is we start to get a nice extension of lifespan. Unfortunately, we don't have a lot of drug, as I mentioned before, so as soon as the controls died, we ended this. We don't know what this might be. We're redoing this experiment because we have little drug. So what we're going to do is we're going to go back and add it at a later time point and follow those out to the end as a way to sort of interrogate this end state. This is significant, though. It's not super robust, but it is significant. And what we might expect is that maybe we'll even see a stronger effect of the drug when we get out to an animal that has an even more impaired motor function, because we have done these proboscis extensions, and we know that even out at this point, if we give the animals the drug, it reestablishes that proboscis extension to the 7-day value. It's just remarkable. Okay. And so we're going to go ahead and do that experiment to see if that's true. The other thing I mentioned is that we do have the drug target. I didn't put this in here, but the drug target has a very interesting, so we did lifespan on the drug target. And the drug target reminds me of the poem, or of the mutant of the drug target, reminds me of the poem One Hoss Shay. Has anybody ever heard One Hoss Shay?
Doesn't one horse
Yes.
buggy?
Yes.
Okay.
Right, so there was William Oliver, I think, was the one who wrote the poem, and he built the One Hoss Shay in a way that it would never break down. And it ran for 100 years until the anniversary of the Lisbon Fire. That's where he was living. And on that 100th day, it died. It fell apart and it looked like it had been milled into nothing, right? The idea was he built it perfect, and it went, and then it all of a sudden just fell. There was no gradual decline in its function, okay? And when we look at animals that lack this S107, and I'm sorry I didn't put it on here, they have a survival curve that goes straight out like that and straight down like that. Almost exactly like that, and no effect of the drug. And maybe that's a mutation where we've maybe somehow revealed something about progressive frailty. Because those animals one day looked fine, and a couple of days later stopped moving, and a couple of days later after that they all died.
And you started out as early age, like a month or 2 months?
In those animals, we did the same thing, where we started the drug right here. So we put the wild type animals and the mutants on it right here. And so they were on the drug for a few weeks. Yes, continuous, in the food.
Did you try to have them up and like just 1 day break, like one and one, and see?
Like an intermittent sort of feeding on the drug? We haven't done that.
And
Like how long it might stay around
one
after we
other
take them
thing
off.
is 107 is a toxic drug that maybe you start with the dose that maybe you need to increase a little bit, you just see the effect of the drug on lifespan, or it's already established that the toxicity of the drug cannot exceed that limit. I'm talking about
No, we don't know that. So what we did is we tried to estimate our concentrations of the drug based on the biochemistry. And said, okay, well, the biochemistry utilizes a certain concentration, and we times it by 10.
And the mutation is in what gene?
It's called calstevin. In us, it's called FK506 binding protein 2. we may rename it. It hasn't been studied before.
So this is hitting the radioiodine receptor.
Yeah, it should be affecting the ryanodine receptor. And this protein, the drug target is expressed in all tissues. So we would expect that, you know, these events could be going on in all tissues. And maybe it's just most manifest in the muscle in terms of the effects on the physiology. But that's one of the reasons. So, you know, one of the reasons I'm talking about this mutant is that we need to go back. And what we're going to do is we're going to take the mutant, re-express the wild type copy in the muscles, and see whether that restores normal lifespan as well as the drug response. To make sure that's true, because that's actually a very interesting sort of survival curve. And one of the things about the behavior is in the behavior, those mutants have reduced behavior at 7 days. It's stable. It doesn't change all the way out until they die.
And do they die at relatively the same time as the, I guess, wild type?
No, they're less.
They're less.
I'm sorry.
Oh, okay.
So if you were to just generate they
Okay.
only live right about here.
I mean, just thinking like, I don't know, this is maybe
It's a box.
philosophical, but like, you want to live longer with some morbidity and disability as you
Yeah.
age, or you want to live shorter and be really healthy the whole time? I mean, that's, I don't know. You know?
Right.
Good point.
You know, so,
don't know.
I mean, one of the things, I think this is a really interesting phenotype,
Yeah.
right? So I think that we have to follow up the survival stuff. We've been thinking of, I mean, right now it's all philosophical, right?
Yeah.
But one idea is this: what would you expect if you remove the target of oxidative damage? What would you see? What would lifespan look like if you took out a major target of oxidative damage that contributed to mortality? I don't know. Maybe you would see, maybe what we're revealing is a second limit to lifespan that is independent of the effects of oxidative redox. These are really cool. That's why all of a sudden I'm getting more interested in muscle.
There's a mutant, animal mutant called suboxide. And why?
Yes. Right, so we can manipulate that stuff. So right now what we're getting ready to do is take the mutants and the wild type animals and give them paraquat. And ask what happens to their behavior and their survival, but also the behavior. Because maybe the mutants will maintain function in the presence of paraquat while the wild type will decline. That's what might be our expectation, is that we will see something that we see now, which is under paraquat conditions, the wild type animals are going to have their motor function decline, and maybe the mutants won't. So maybe they'll be resistant actually to paraquat. We're doing that experiment now. So in the end, this is the end of what I was going to talk about today. So then maybe motor function is a really important age-dependent morbidity in Drosophila. That seems so like no kidding, but it actually hasn't been shown. And so we think we might have the way of actually, with this type of an experiment and doing the mutants and the rescues, we might be able to really make a strong case that, you know, one of the reasons flies die is because they have lack of motor function, probably affecting something about how they eat or feed.
Can you measure the size of the muscle fibers?
I think we can.
I mean, is the lean mass increasing as it flies?
Yeah, right. You know, oh, God.
I know, it's
Fly
tough.
physiology.
It must be really small,
I mean, I think we could, I mean, you're right, the difficulty is can we find the muscle? I think we can do everything, because we've done EM on different muscle groups. It's just, we've never tried this. I think we can do it. I mean, one of the reasons I picked this muscle group was because it's amenable to this type of stuff. I think we can find it. We know where it is. We know exactly in the head where this thing is. And I would expect that we should be able to get back and section these things and do HE staining or something very simple to look at muscle fibers. I think that's absolutely the way to go. We have done counted muscle fibers. And so, you know, interesting, different than the mammal is there's no stem cells, right? So there's no satellite cells here. And so there's no replacement of fibers. And actually, when the animals get to about 60, they almost always have lost 1 or 2 fibers by that time. And so, you know, what we're looking at has nothing to do with replacement. It's all about fiber function. Right? So at some levels that may be a benefit for us because we don't have to worry about the confounding sort of aspect of diminishing stem cell replacement or something, that these are the fibers that these animals we think are born with. And so what we're monitoring is how that fiber function is maintained with age.
Did anybody look at if S17 effect on the total level of ROS generation? Did you look at that?
No. Well, yeah. So the data out there would suggest it doesn't have anything to do with generation of ROS. So the S107 doesn't affect the ROS generation. It simply reestablishes that interaction between the ryanodine receptor and that calcium protein. We have not done those experiments.
So, yeah, ROS is not involved in this part.
Aren't mutations in that receptor one of the causes of malignant hyperthermia?
In the ryanodine receptor? Maybe.
Yeah. No?
That might be true.
Is there a
That sounds
someone taking
familiar.
the board?
Doesn't ring a bell.
No.
Well, we know that ryanodine receptors
Do you know
function in
which-
neurons as
do you-
well. So
I thought there was a role for it, yeah.
In malignant hyperthermia?
I think that's right.
yeah,
What happens to a dietary restriction? Does that
we haven't done it.
change the set point with age?
We haven't done it.
Nothing?
We haven't done that yet.
Okay.
You know, I would expect that it would delay it, but I think it's still going to happen. But that's an experiment I'd like to do. Just need more people. But we don't know about the ADF, and you know what we're trying to figure out what that is, and what we want to do. And so I have a postdoc here, and he's going to. So what we are doing is we're FACS isolating out motor neurons from the fly. So we have a pure population of neurons, and we're going to do a sort of longitudinal microarray analysis with the idea of hopefully, we think it's a stable change. So we're expecting that there's going to be a change in gene expression, and hopefully by looking at the gene expression, we can get lists of co-regulated genes that might tell us what transcription factors are driving this change, and that might be a way for us to get into the age. What age is doing, how age is turning on this change. And so we are trying to get funding for that. But and I'd like to do the DR experiment, just because I'm expecting it would delay. But we don't know.
It'd be interesting to see anyone has given it to humans.
I have not seen it. I think this. I think this drug has some potential.
Cyclists.
That's it. And
And
then
all the cyclists before and after.
and you know what? Those guys are all about. They're all about results.
Yeah.
You know what I mean?
They would be happy to be subjected
Right.
to all kinds of things, I would guess.
But you know what? I really haven't looked further along that. I do know that Andy's involved with the company, and that there's been some data coming out from them that have shown some central targets. So that does look like the drug is affecting nervous system function as well. And there's a paper about it treating some, I think it's Parkinson's model of a mouse. Fairly recent. Yeah, it's a pretty it's pretty remarkable. And I don't. Like I say, I think it's kind of interesting. We might be able to make some headway into some of this. For us, we're interested in teasing out this sort of survival lifespan,
It might be
philosophical sort of ideas on
Could it be making the muscle secrete something
Well,
that's going to other tissues and make the animal live longer?
I would say that if you improve muscle contractility and you improve muscle health, then you have the opportunity to be secreting more BDNF, NGF, right? I think that BDNF in the blood, I think a lot of that comes from muscle. So yeah, I think they're important organs. Whether or not S107 directly, I don't think so. I would say that if it's stimulating the release of something, it's doing it because it's improving the overall contractile sort of apparatus and health of the muscle. When we add it to synapses, so I showed you when we saw a little deficit in release, we see that as well in
in larvae, and so we suspect that maybe there is a little bit of a change in release, and that might be to something about calcium handling by ryanodine receptors during release. We don't know anything about that, but I don't think there's any evidence that would suggest it's stimulating a release event other than just by secondary effects of improving muscle function. Oh, God, I cut off all the important people. So Rebecca is the person who did all of the recordings pretty much that I showed you. Tabby did all this behavior. Jorge is a new postdoc who's getting thrown into this, and these were Leo, Denise, this is Yamin, Joel. Joel was the former postdoc who really gets a lot of credit for driving this. Well, I would say I was running the whip. He was doing all the hard work. I would say maybe I was driving him, but he was the one who really worked through all the hard stuff to get the recordings to work. And these are obviously some collaborators that we have on campus and some of the funding that we've got.
Thank you so much for coming. Be sure to sign in. I got a head count of I got a head count of 14.
Yeah, no problem.
So you're okay with the mark?
Yeah. I'll implement back the
Each one's in the door.
Sorry about that.
I can happy to do because I have to cognition expert, but
Yeah, helped him.
and I guess I'm decently expert in physical function. I mean, geriatric assessment and measures of disability and
So I'll tell you how I interpret it.
frailty
Because
type measures.
and this was something that took me a while to try to come about, which was, we get that